Students, let's start a new chapter today, and the name of the chapter is Biotechnology Principles and its Processes. Now, let's at first try to understand about the term what is biotechnology. The term biotechnology, it deals with techniques of using live microorganisms or plants or animal cells all or their components or enzymes from organisms to produce products and processes. That means it infers services that is useful to human being. So what does this definition says about the term biotechnology? See, biotechnology that was in use for many decades, or you can say that it is referred at once upon a time it was referred to as kitchen technology. Now let me give you an example how. Here bacteria, as you know, can convert milk into curd. Bacteria, by its process which is called as fermentation, it can coagulate the protein in the milk, and thereby we get a product which is called as the curd. And we have been using curd for many years now, and this example give you an idea about how this biotechnology can be. From ancient times, many microbes they were used in different ways. Say for example, for the production of food, sometimes for the production of beverages, organic acids, vitamins, etc. Now, the living organisms they were employed, and here I have given the example of bacteria or their components, and that produces some products or they are processed, and these products and processes they are useful to us. But this biotechnology or this definition of biotechnology, we can refer to it as the old biotechnology. Biotechnology, it has taken a different turn now, or it is seen with the wider aspect. Therefore, biotechnology, it is also called as the manipulation of organisms or cells or parts of cells, molecules for generating useful products and services. So that is the manipulation of the organism. And now, therefore, the definition of biotechnology has changed according to European Federation of Biotechnology. So from the test, you can have the definition that was provided by European Federation of Biotechnology, and it is stated there that it is the integration of natural science and organisms, cells, parts thereof, and molecular analogues for products and services. That means now it is a integration of the natural science. So here chemistry will be included, here mathematics will be included, related to the applied biological aspects such as biochemistry, molecular biology, genetics. All such aspects of biology will be integrated together, and that integration that will lead to the use of organisms or a cell or a parts of cell or its molecular analog. such as the DNA and the RNA for producing products and services which will be useful to the human being. So, this is the definition of what biotechnology means to us. Now the term biotechnology, it was first coined by a Hungarian engineer called as Karl Ireki in 1917 when he was making some breeds of pigs, and there he employed biotechnology, and therefore the term biotechnology, it was coined by the Hungarian engineer called as Karl Ireki. Now let's come to the principles of biotechnology. The principles of biotechnology, it includes genetic engineering and bioprocess engineering. And in our syllabus, only the part genetic engineering is discussed in detail. Now let at first try to understand both the principles of biotechnology. So in biotechnology, the first principle employed is genetic engineering. Now what is genetic engineering? So it is a technique to alter the chemistry of genetic material. As you know, the two genetic material or the nucleic acids, those are DNA and the RNA, to introduce this into host organisms and thus change the phenotype of the host organisms. So in this definition of genetic engineering, it is said that the science that deals with the manipulation of the DNA or the RNA, so that is manipulated, that is changed to improve the quality of organisms for specific purposes by implanting desired genes, and in that it changes the phenotypes of the host organism. So that is what it is said about the term genetic engineering. So in biotechnology, in modern biotechnology, the alteration of the DNA material that is introduced into the host organism. That means here two organisms will be included. One from where the desired gene will be brought, and the one where it will be introduced. The one where it will be introduced, that is called as the host organism. So it is introduced into the host organism, and thus it changes the phenotype of the host organism. So this is genetic engineering. Why it is called as genetic engineering? Because here the genes are included. And the other name of genetic engineering, it is also called as recombinant DNA technology. Now let's come to the second principle. And here the second principle that biotechnology employs is bioprocess engineering. So what is bioprocess engineering here? As you know that here live organisms are required. For example, bacteria, fungi, etc. So the maintenance of sterile, that is microbial contamination free ambience in chemical engineering processes to enable growth of only the desired microbe or eukaryotic cell in large quantities for the manufacture of biotechnological products like antibiotics, vaccines, enzymes, etc. As the live organisms, either in the form of a microbe or it can be any kind of eukaryotic cell, as they are cultured within the laboratory, and in the laboratory a maintenance of a sterile, that is microbial contamination free, there shouldn't be any kind of bacteria or fungi or such kind of microorganisms which can contaminate or damage, destroy the culture medium. So such environment has to be created by applying chemical engineering, so that the desired microbe or eukaryotic cell that can be grown in large quantities for the manufacture of our desired biotechnological product. And those biotechnological product, it can be either antibiotic, vaccines, enzymes, etc. So this genetic engineering and bioprocess engineering, they are used as a principle for the purpose of obtaining biotechnical products that will be useful for us, the human beings. I hope you have understood the introduction of biotechnology. So till now I have spoke about the term biotechnology. So biotechnology, it simply means the technique of using any kind of live organism. It can be any kind of eukaryotic cell from plants or animals, or any of their components or enzymes, which is useful for some other organism, that is the host organism, to produce some products, or it can have some processes such which is useful to the human being. And there I've said about two types of biotechnology. That one is the old biotechnology, which is also called as the kitchen technology, where the microbes, they were used to make products, different products, such as food, such a card, different type of beverages, organic solvents, etc. So they were used in order to obtain such kind of product. But now biotechnology, it has taken a different turn, and it is seen with the modern and broader aspect, where the manipulation of the cell, either in the genetic level, is generally employed, and therefore modern biotechnology generally deals with such kind of manipulation in the gene level, and that is now the main aim of biotechnology. And there we discussed about the principles of biotechnology. Those are genetic engineering and bioprocess engineering, or that you can call to be chemical engineering. And now let's discuss about the first principle, that is genetic engineering, in details. Now let's have some more idea about genetic engineering. Genetic engineering, or recombinant DNA technology, and in short it is also called as rDNA technology. So as I have said, genetic engineering, it is a kind of biotechnology which deals with the manipulation of the genetic material, that is either in the DNA or RNA, by man in vitro. That means artificially the DNA or the RNA, that is manipulated according to the wish of human beings, or the one who is responsible for conducting such kind of researches. And it is called in vitro because that is done within the lab, outside the cell. Now, how this genetic engineering, it became one of the important aspects of biotechnology. So genetic engineering, with two main discoveries, that became the important aspect of biotechnology. That is, when plasmids, or the use of plasmids in bacteria, were used for the purpose of replication in the biotechnological studies. So as you know, the plasmid, this is a specialized circular DNA that is found in bacteria. And this plasmid, as they are made up of short nucleotide sequence, or generally less nucleotide sequence, having different point of origin of replication. So they can undergo replication along with the independent of chromosomal DNA. And this particular discovery for the use of plasmids, that led to a new dimension or addition of a new part in the whole process of biotechnology. And then came the next discovery again, that is about the use of restriction endonucleases. Now, the restriction endonuclease, these are enzymes that are used by the bacterial cells for cutting unwanted DNA segments from the bacterial DNA by themselves. So they naturally employ this technique of using restriction endonucleases. And this nucleases enzyme, they are used, or they are a type of enzyme that are generally referred to as molecular scissors or biological scissors, because they can cut the DNA segment. And this restriction nucleases, in a study by Arber, Nathan and Smith in the year 1970, they could use the enzyme in the biotechnological research or in the genetic researches. And they were awarded Nobel Prize in 1978 for this great discovery, which can break DNA at specific sites. So, this was the next important discovery in biotechnology, or we can say in genetic engineering. So these two discoveries led to some new innovative works in the field of biotechnology or in genetic engineering. And in 1972, a genetic engineer called Paul Berg, and Paul Berg, he is regarded as the father of genetic engineering. He tried to introduce a gene of a virus named SV40 into a bacteria with the help of lambda phage. That is a virus again. And he was successful to transfer the gene of SV40 virus into the bacterium with the help of the vector called lambda phage. And in that way, Paul Berg, he was awarded Nobel Prize in the year 1980. So in that way, genetic engineering, it became a new part and a very important part of biotechnology. And later in modern biotechnology, it became a very important aspect or important principle for manipulating or for adding new characteristics in an organism just by changing the genetic aspects or changing the structure of the DNA according to the wish of human being. So this is a small introduction for the term genetic engineering. So next we are going to study about the different tools or different you can say the requirements that is used in the genetic engineering study.